Hi, I'm Irene Ng. I'm the chair professor at the Department of Pathology and also the director of the State Key Laboratory for Liver Research of the University of Hong Kong. I'm leading a group working on the molecular pathogenesis and molecular pathology of liver cancer. Hepatocellular carcinoma, abbreviated HCC, is a common cancer and a leading cause of death worldwide. There are many challenges in liver cancer. It creates a huge healthcare burden. There is also social impact as patients are at the prime age of their life. It is a deadly cancer, and patients with HCC have poor survival rates. 80% of the HCCs are inoperable. Even when the tumors are operated on, there is a high tumor recurrence rate. The tumor is resistant to conventional chemo drugs. The FDA-approved molecular drugs have modest effects. Significantly, the molecular drugs are given with no patient stratification, that is, on a one-size-fits-all basis. In this regard, we have recently performed a study to find biomarkers for molecular stratification of human HCC using genomic analysis, which combines exome sequencing and deep targeted sequencing, focusing particularly on the mTOR pathway-related genes. mTOR, which is mammalian target of rapamycin, is an important signaling pathway controlling a number of significant cellular physiological processes converging onto cell growth. So what are our new findings? Using high-throughput next-generation sequencing, we performed the mutation screening and observed there are frequent mutational disruption on multiple key components of mTOR signaling pathways. In particular, tuberous sclerosis gene, abbreviated TSC. So TSC1 and TSC2 are the two most frequently mutated genes in the mTOR pathway. The overall mutation rate was 16.2%. They collectively define a novel molecular subset of human HCCs with more aggressive tumor behavior. Therefore, mutation-dependent mechanism is likely to be another major cause leading to mTOR hyperactivation. Furthermore, we use patient-derived tumor xenograft for drug testing. These PTDX are TSC2 mutant models, which are therefore biologically relevant. We demonstrated the therapeutic options and benefits of hypersensitivity towards rapamycin treatment. So how might it impact on clinical practice in the foreseeable future? In sum, TSC1 and 2 mutations represent one of the most frequent single categories of genetic alterations found in human HCCs. These mutations will lead to mTOR hyperactivation, which define a novel molecular subset of human HCCs. It is likely that they are actionable through inhibition by rapamycin or its derivatives. Our study therefore signifies personalized therapeutic option for a novel molecular subset of patients with susceptible mutation in their HCCs through the inhibition of mTOR signaling. Thank you very much.